Hello, everyone. Good morning, good afternoon, or good evening, wherever you're joining us from. I'm Ara Khachaturian on behalf of the Campaign to Prevent Alzheimer's Disease, the Brainwatch Coalition, and the International Neurodegenerative Disorders Research Center. Welcome to episode 5 of our podcast series, Empowering Proteomics and Transcriptomics in Alzheimer's Drug Discovery. Today's episode is titled, The Word is Data, Data, Data. Creation, mining, analysis, and interpretation. Are we getting closer to precision?
And I am Ornit Chiba-Falek from the Division of Translational Brain Sciences in the Department of Neurology at Duke University. As always, we want to thank Frontier in Molecular Mechanism of Neurodegeneration for their generous support in helping launch and sustain this series. Across this series, we have been exploring how we can translate omics discovery and implement omics technology, especially proteomics and transcriptomics, in the design and performance of the next generation Alzheimer's disease clinical trials. We began by looking at what we have learned from landmark study like A4 and AHEAD. Then we explored how biological mechanisms, such as the mitochondrial cascade hypothesis, intersect with precision medicine. And in our last episode, we took a deep dive into leveraging omics-based endophenotype in defining Alzheimer phenotypic heterogeneity across diverse population as the next step in advancing precision medicine in Alzheimer's disease.
Exactly. In our last episode, we specifically discussed how plasma biomarkers based on molecular endophenotypes can improve Alzheimer's clinical trial efficiency and diagnostic accuracy, which links directly and builds off of our 2024 JPAD editorial. Today, in episode 5, our theme is focused on when the technology tail wags the scientific dog. We are hosting today one of our editorial's co-authors. We're going to take a step back and ask a different kind of question. What happens when we lose precision, not in the assay, but in our thinking? We use terms like endophenotype, biomarker, or even Alzheimer's and related diseases as if they mean the same thing to everyone, researchers, clinicians, trialists, sponsors, but often they don't. That misconception creates a real challenge in trial design, endpoint selection, and regulatory alignment.
And beyond the terminology, there is an issue that is even deeper. Are we still asking good questions? That will guide us forwards? Or have we let technology, the platform, the analytical pipelines, start driving the science rather than serving the science? That is why we are especially excited to speak with Dr. Lutz, who brings clarity, structure, and real-world insight to this challenge.
Dr. Mike Lutz is professor of neurology and pathology at Duke University. He co-leads the data management statistics core at Duke UNC ADRC and has spent his career designing computational tools from basic research applications to trial infrastructure, such as that Alzheimer's research is more precise, more translational, and more biologically grounded. From CSF proteomics to biology of aging, machine learning to neuroinflammatory biomarkers, Mike has helped to build the platforms, but also to question their limits. And beyond that, before Duke, he led the computational biology team at GlaxoSmithKline, giving him a rare perspective on both academic and industry science. Mike, welcome.
Thank you, Ara and Onit. I'm very pleased to join the discussion today.
Well, we're very happy to have you. So we're going to start off with study design, cohort logic, and the mirage of GWAS simplicity. And starting at this foundation with how we design our cohorts and define our samples. You've worked on some of the earliest efforts to shift away from case control simplicity, including your work with Tomorrow, a study that was one of the first to use genetic enrichment strategies to identify individuals more likely to be responsive. Also, looking at conversion from MCI dementia as endpoints has been a new measure the Tomorrow trial introduced. Looking back, how do you assess those early considerations in trial design? What did they get right about personalization? What did they miss about the complexity of Alzheimer's heterogeneity?
Sure. So the Tomorrow study showed some quantitative results on the precision of age-related risk for conversion from MCI to dementia, specifically in a 5-year time frame, which is really important in the context of design of clinical trials, which may be time limited. The very distinctive survival curves that we drew based on genotype showed how two specific genotypes shift these curves right and left. And you can envision how during the course of a trial, someone who enters at a given age and then is randomized into the different groups would sit on these curves. Risks to convert from MCI to dementia in 5 years was really critical because as is really the concept of age-related risk. For personalization, it was a strength that the trial was designed to test both the biomarker and the drug. So although we had randomization at both steps, the trial could complete with a number of different outcomes. The drug could be viewed as successful or not. The biomarker could be viewed as successful or not, and both could be. In both cases, you obtain quantitative evidence around the use of this biomarker to enrich for individuals that are likely to convert from MCI to AD. On the composite, it's interesting. That composite was tested and calibrated in different ethnicities, and that's another important aspect of heterogeneity is understanding the impact of ethnicity on specific genetic biomarkers.
Thank you, Mike. And building on that, we have all worked with data set from genome-wide association studies that treat Alzheimer as a binary trait, case control study design. But the binary framing doesn't reflect the true biology and doesn't capture the real disease heterogeneity, the actual clinical, pathological, molecular subtypes of Alzheimer's disease. What do we need to do in core design, endpoint selection, or inclusion criteria so we can capture the real diverse pathways and heterogeneous subtypes that define Alzheimer's disease as it's actually a spectrum of diseases?
Yeah, that's a really good question. The genetic biomarker that we used in Tomorrow was based on two genotypes. So in a sense, you could consider it a rough cut for precision. And then really introducing this concept of age-related risk for the genotypes was important. You could use SNP panels and perhaps polygenic risk scores to show some increased precision, perhaps for specific age ranges. I think the real increase in precision, though, would come through the use of dynamic biomarkers, assuming you could obtain the data needed to calibrate them. And that calibration would have to reflect some of the different heterogeneous conditions that you would be looking at. There could be heterogeneity based on sex. There could be heterogeneity based on other comorbid conditions. All of these things, the calibrating these dynamic biomarkers, I think, is key. Now, the dynamic biomarkers also probably have somewhat more complexity to them than, say, a SNP panel. The SNP panel is determined basically at birth, and we can identify which SNPs are predictive and could be used, say, in a tool like a biomarker risk algorithm. The dynamic biomarkers have to really be considered for really if they're fit for what we're trying to measure at the end of the trial or during the course of the trial. So you have to really consider the longitudinal design of your study, rates of change, any information that can go into the initial planning of the study, including powering the study, is really important to find out. And I think these dynamic biomarkers when they're measured over time, and we're starting to get this type of data now, would be very valuable to untangle disease heterogeneity.
Thank you, Mike, for sharing your perspective. And I would like to shift gear now to a topic that focuses on method and whether method becomes the mission and renaming the tech curve. So let's talk about the speed of change. You have lived through the shift from bulk to single cell omics, from short reads to long read sequencing, from fixed pipeline to real-time analytics. That rate of progress is thrilling, but is it distorting? Are we seeing technology drive the research direction rather than the other way around?
Right. So the technology certainly enables measurements at an unprecedented scale. And I think scale is sort of the operative word to use in response to your question. We can literally now get down to the individual of measurements at individual synapses. So single nucleus omics lets us consider cell type specific changes that map to a level of biochemical precision that would have been hard to imagine even just a few years ago. The analytical process, though, needs a corresponding reset in order to increase precision. So many studies still progress with analysis in a very linear fashion. You start with the variant, you go to the gene, you go to the pathway, you go to the association, some kind of an interpretation, and publish. We have to move beyond that. We have to move beyond this linear type of process of doing our analysis. Indeed, probably, or perhaps instead, you can envision a network of different data types and analytical tools where the nodes are the data. These could be omics, genetics, transcription factors, isoforms. Some of these are quantitative, some of these may be more qualitative, and edges or analysis approaches. So the network is ideally built to address a very well-formed research question.
Thanks, Mike. That's really compelling. And just sort of thinking just back to your previous answer on dynamic biomarkers, if we want to get a little bit deeper into this topic, and I love the topic about sort of the nonlinearity and the complexity. So let's bring in the question that's related to all of this. What does it take to build a research culture that, stays adaptive to technical change, but doesn't sort of lose sight of its first principles? In your experience, where have academic centers and industrial sponsors get this right, or where they maybe are falling short?
Okay, so I mean, a research culture adapts to technical change, has to balance really between two poles. Researchers need some time and encouragement to try new methods and technologies. So I always find a few new approaches or software to try after browsing the posters at conferences. Many of these don't pan out to very much. Some of them become part of analytical pipelines. At the same time, many of the primary research questions don't change that much. So you need to be cautious and not develop centers or programs or initiatives with aims that are not well defined in terms of what the impact will be, in terms of what the impact would be, say, on clinical trial design for one specific trial, for many trials, for, say, statistical analysis approaches that are used in trials. Some new research questions will surface, and analytical processes will have to be updated and adapted, and that's fine. It's just that the process really needs to be done thoughtfully. And in industry, there's usually a pressure to get things done very quickly, and that can lead sometimes to really not having the kind of careful evaluation of the new technologies that are needed. Yet industry, one thing that industry does quite well is scale. So they can scale up and test hundreds of thousands of compounds rather quickly. They can synthesize different compounds. So you can explore chemical space and biological space at a scale that's quite high in industry, which and we may cover this a little bit later, but some of the industry academic collaborations can be key. So industry does scale really well, Perhaps you can say that academia perhaps does depth well. In academia, you're going to be more focused on really a deep understanding of mechanism. Many of the studies that are done in academia really want to go deep in terms of understanding mechanisms and targets. Industry, yes, they want to know the target. They want to get to the target, and then they want to optimize compounds around that specific target. The in academia, more understanding, well, how does that target fit into the context of other systems? So systems meaning starting from, say, a receptor to the corresponding signaling pathways, and then signaling pathways perhaps to the level of an organ, and then eventually to the level of the entire physical system.
Great, thank you. So we're gonna shift gears and move to our next corner, which is on data harmonization, and not just another technical challenge. And certainly, Mike, you've been a leader in building data infrastructure across all sorts of cohorts at the ADRC and beyond. And we often hear harmonization discussed like as a software issue. But how much of the challenge is actually about language, assumptions, or even philosophical differences?
So it's a good point about the harmonization. The challenge is usually about the language, definitions, and decisions. What I'm getting at is that there's a myriad of decisions that are made when you're analyzing a specific data set. So and there are several efforts, very large scale efforts, to harmonize across multiple data sets and multiple institutions. Um, and we can look at protocols, we can see how things are done. In the laboratory culture, there's a lot of emphasis on putting sufficient information in a protocol that someone in another lab can run it and obtain the same results. We have to have that same level of precision and accuracy in our computational work. We have to understand what are the parameters, and that gets more and more complex when we start bringing in, say, machine learning and AI algorithms that have various parameters that have to be tuned. Some of the technology is helping us in that way in terms of reproducible research initiatives that record every step that you're doing when you're analyzing a data set so that another analyst can reproduce those steps. But again, it really is more about the language, the assumptions that are involved in a particular study. So I think thinking about harmonization, it does always help to think about the harmonization at the start before you collect the data. It's much more difficult to do it after you have a huge data set. But again, I think it's it's not only the data dictionaries, but thinking about how the analytical process would so when you're trying to analyze data together, you can indeed combine these data. You may want to be comparing across different platforms. For example, right now with proteomics, there are a number of very popular um, platforms out there. So you want to make sure, again, that if you're going to try to harmonize data across the platforms, you understand what the limitations are and what the assumptions were for that platform.
Thank you, Mike, for your insightful response. And I would like to continue on the topic and ask you, what are the practical steps researcher and funding agency can take to avoid mismatches at the point of integration, where one team's definition of phenotypes or diagnosis is defined by another team as a noise or a reversible change? How do we create infrastructure that is not just technically solid, but also conceptually coherent?
Yeah, avoiding mismatches at integration points is challenging. You can use guidelines, standards, and ontologies. These can be helpful, but they need to be considered at the start rather than a retrofit. So there are some good learnings from, say, the National Alzheimer's Coordinating Center Uniform Data Set. So trying to integrate data from all of the ADRCs is a particular challenge and something that the NAC, the National Alzheimer's Coordinating Center, spends a considerable amount of time thinking about. They are now working to expand into the omics world as well, which I think potentially can have uh, a great strength for the field if indeed we can rely on an organization like NAC and particularly the resources of NAC to create these integration points for some of the more diverse data that we're starting to work with and the more complex data.
Thank you. Thank you very much, Mike. And I would like to change now the topic and focus on discussion on collaboration or what making collaboration real across disciplinaries, from trialists to statisticians. You are someone who lives in the middle of translational teams between clinician, trialist, experimentalist, and data scientist. What are the most common misalignment you see between those groups?
Sure. So misalignments result from differences in experience, background, and motivation. Statisticians are experienced at advising trialists and experimentalists in designing rigorous studies. To do that well, though, requires understanding aspects of the research question, which may be quite subtle. So, indeed, having the research question and formulating and honing the research question together is a great way to minimize misalignment. People really need to outline what their assumptions are and what they're thinking about. So sometimes a statistician needs to make a map from a quantitative result to a more qualitative result to putting it in a clinical meaningful context. For example, what does a 2% change in some measure mean in terms of cognition or in terms of quality of life? That's how clinicians tend to think in terms of, well, how is this going to make a difference for the patient? Will it make a difference in the quality of their life? Will it make a difference in how they interact with people in terms of what they can do? Whereas when you're thinking about the statistical result, well, it's significant and maybe it's a small change or maybe it's a large change, but how do you map that to something that's clinical, meaningful, and impactful? And that's really where the shared understanding comes in, is what that impact is. Um, Clinicians and experimentalists on kind of the flip side, need to think very carefully about sources of variability and heterogeneity in addition to testing a primary outcome at, say, p equals 0.05. So, to them, the statistical test can be very black and white. Well, yeah, the test result may be very black and white, but until you really start to think about variability, potential confounders, all of these things, I mean, sometimes it helps to have a whiteboard and just kind of say, all right, what are all the possible sources of variability that we have in this study? And what could they mean? For some studies, Bayesian methods may be most appropriate, and then it's going to be incumbent on the statistician to explain how Bayesian works, how Bayesian methods work. Now, for observational studies, it's helpful to try to match the structure of a randomized clinical trial to the actual structure of your observational study.
Thank you, Mike. And I'm sort of reflecting on your answer, which was really interesting and great. We all have spent time editing journals, and one of the things that always sort of bubbles to the top is personality and communication style. And certainly, what are the structures that help collaboration work? I mean, you talk a little bit about the certain shared assumptions, but in terms of the timelines and vocabularies, what else do teams need to agree on from day one to be super effective?
Right. On structures to help the collaboration work effectively, starting at the beginning of the study is really key. Too often, the analytical folks and statisticians are brought in at the end. But if you can start at the beginning, that's where the clarity around the research plan develops. You can think about planning meetings for the collaborators with specific deliverables and timelines. That helps to put things in terms of a project context that they can work with. More granular deliverables with shorter time frames at the start of the study, I think is important. then as the team becomes more comfortable working with each other, with sharing their results, with how they describe their results, then the deliverables can be somewhat less granular and the timeline's a bit longer and more, a bit more um, more challenging, perhaps. Um, so common vocabularies, that's, also an interesting idea, but I'm not entirely sure how you implement it. I think background information and prior experience or publications or studies are helpful to start. And statisticians may want to share some specific, I mentioned Bayesian, for example, sometimes sharing information on how Bayesian analysis is done is helpful to a clinician that may not even remember kind of the Bayes theorem from their undergraduate days. Statisticians need to have very simple, clear explanations of concept available, things like variability, distributions, why you might want to use, say, non-parametric statistics or say Bayesian statistics. And then I think, you know, moving forward, a review of the methods and results regularly really are what make for an effective collaboration, so that the analyst or the experimentalist kind of doesn't go down a pathway for an extended period of time, and then say the analyst or the experimentalist says, "Oh, well, by the way, you missed this one key assumption." Again, I think regularly reviewing your methods and results is really quite key.
Well, thank you, and I feel like Mike, you're being very, very modest. So for people, Mike, I think is an absolute master at in terms of sort of re-engineering and thinking about how to almost where you want to go and sort of build backwards from there. And I don't know, maybe that's a result of your experience both in industry and academia, but in terms of setting milestones, mindsets, and missions, I mean, definitely you've been on both sides of this. And we've talked about how different the milestone models are both in industry and academia, where industry will often ask what's that minimum evidence needed to advance a candidate technology, and academia often asks what's the next interesting question. And so from your perspective, how do these different mindsets impact, for example, biomarker development or clinical trial design and translational science?
Yeah, the milestone models for academia and industry are quite different, as you say. Academics often operate in the time frame of a 5-year grant with rough milestones and relatively independent specific aims. So there, the endpoints are designed, success is defined, the steps are defined, all focused around, say, somewhere between three and four specific aims that kind of will drive a work plan for 5 years. Industry kind of has a few different scales that they work on. Some are quite short and some are quite long. So industry, at least the pharmaceutical industry, has again this traditional linear chevron diagram of drug development all the way from discovery to phase 3 and eventual um, uh, approval of the drug, which can be a long multi-year time scale. Um, and I think what you have to overlay on that are the go, no-go decision points. And some of those, the time scale in industry can be very short. They can be just a few weeks to identify whether or not something is really just not worth pursuing any further. And I think it puts quite a bit of pressure on the teams to have to deal with some of these go, no-go points before they've really had time to adequately get the data they need to gather the information necessary to make a good decision. For biomarkers, um, perhaps a bit more emphasis in academia on understanding mechanism and interpretation. So for them, for the academics, really understanding the context of of the biomarker in terms of human biology is important. Now, for industry, it's sort of what the application would be. So is the biomarker being considered for a companion biomarker, say for safety or efficacy? And then the question is, well, how well it would do. So in a sense, you take your candidate biomarker, and the biomarker may be a single molecule, or it may be a panel, or it may be an algorithm, or it may be some combination of all of them. And some of the performance evaluation criteria are quite similar for both. And that's fine. You may evaluate your biomarkers, the accuracy of your biomarker, the precision in a similar way. But the industry is really going to be looking primarily if it's fit for purpose. Does this biomarker, is it going to help with ultimately the drug that they are that they are looking at? Does it mark specific safety problems, like individuals who should not take the drug? Is it a marker for efficacy? The individuals in this group will particularly benefit from the drug. And then if you take it to the clinical trial, which I think is again a really interesting consideration, can this biomarker help for the clinical trial for pharma? They're going to say, well, if it helps, make a go no go decision earlier in a clinical trial, great. If it helps identify individuals most at risk and then most likely to benefit for the drug and most likely to contribute valuable data to the trial, then yes, then it's fit for purpose.
This is a very important reflection, Mike, that you bring from both of your experiences in these two different environments. And I would like to extend on this direction. Is there a path forward that merge the strength of both clarity of purpose, defining major goals, and methodological rigor, while circumventing the pitfalls of both academia and industry cultures?
Sure. So how to merge both is a great question. There are successful examples of industry academic collaborations. Kind of as you said earlier, industry has access to incredible scale in terms of chemistry, testing, and databases. Unfortunately, industry is really late in the game to data sharing, which could circumvent a lot of pitfalls and I mean, even in terms of better design for experiments. Industry has been very reluctant to put data out there. There are some good examples where it has worked. For example, AMP-AD is an example where there are successful collaborations between industry and academia. But still, when you think about the data that's available on AMP-AD compared to what's available, say, in a major pharma company's chemical database, structure activity relationship databases, that kind of data, if the academic collaborators had access to that scale of data, it would really greatly improve the tools and maybe more importantly, even just not only improving the tools, improving the applicability of the tools to actually have an important impact. Recognition, sort of the strengths and resources of each collaborator is critical, as is setting clear goals and milestones. Expectations and reality can be quite different. So sometimes you build on something to build on is that both academia and industry have cultures that encourage multidisciplinary teams. So industry, again, in pharma, they've kind of gone from some years ago, chemistry and biology, then bringing molecular biology on board, bringing eventually some of the omic technologies on board, bringing in computational chemistry. Multidisciplinary teams now are very much the way that pharma work. Likewise, in academia, we've gone from siloed labs where an individual kind of works at the same question or problem for most of their career to, again, multidisciplinary teams and collaborations. So that aspect of culture is sort of starting to merge between the two, and that really helps improve the methodological rigor since there can be an emphasis on strong experimental design and analysis plans.
Thank you, Mike. And I would like to make now a transition and bring to the stage the black box of AI and extend the discussion even beyond the black box and see how we can make AI accountable in omics driving research. And Mike, maybe we can start with this corner with a bit of a timeline. Could you walk us through how analytical tools have evolved over the time axis in your world, from the early days, from classical biostatistic tools like JAMP, I know that you like JAMP, or basic regression models, to more recent layers of machine learning, and now into this very broad, sometimes even vague label of AI? What is the change in terms of what this tool can actually do? And just as importantly, how we can interpret them.
Well, analytical tools have changed considerably since I started out 36 years ago, and I could start the timeline now instead. Um, Analytical tools in the earlier days were usually handcrafted programs, or sometimes some general purpose statistical software tools. You mentioned JUMP, and the advantage to JUMP was being able to visualize data, and that continues to be important. Visualization is still very key, whether it's a small data set or a very large data set. But we go far beyond now what an individual human can interpret just visually. I do want to highlight R because I think R has changed the landscape, primarily not so much that it's the best software, but what it is it's created a vast community of resources for both statistics and bioinformatics. And it's this community that's built up around R that's the real strength of it, and how, again, I think the analytical landscape has really changed a lot. So rather than sort of an individual scientist writing their own code, say to do curve fitting, and then bringing that up, now there's more than likely an R package out there that's close to what you need. Now, as always, you have to make sure that it's statistically rigorous and it performs for your specific application. But you probably don't have to start from scratch. You can ask others for help. That's helpful too with a lot of the communities. So I think that, again, I think shows some of how this whole set of evolution of analytical tools is happening. It is becoming more community science this way. Um, Machine learning and AI, that's a good question too. It's challenging to consider if statistical theory and applications are keeping up with the scale and the details of the new technology, like single nucleus transcriptomics and proteomics. That's important. Large numbers of analytes can present a challenge for false discovery rate methods. And again, as we mentioned earlier, sources of variability. Now we have to think not only at the sample level, but also the cellular level for single nucleus transcriptomics. They needs to be considered. Sharing best practices is critical, and there's always been some approaches that are black box. While these may be appropriate in some settings, generally they don't have applicability in clinical or biomedical applications, although there are some notable exceptions.
Thank you, Mike. So I'm sort of sitting here thinking a little bit about this, in terms of the timeline, and when did we cross the line, or maybe we haven't yet, but when do you think we could be at a point where we can stand on a stage at a meeting and show a proteomics and transcriptomics result and say, we're using AI here, and that be taken seriously? Has it happened yet, or what needs to happen, perhaps methodologically or culturally within the research community, to make such a claim credible?
Right. So many fields are trying to understand how to place AI to make a clear, credible statement that something is the case, that it can show really clearly stated results that hold under a range of conditions that represent the target. So it could be that individuals of a certain stage of a disease, maybe it's credible for that. Could it be that some biomarker addresses disease heterogeneity or comorbidities? AI could fit in as part of the methodology to get in, to get in to as an endpoint. But in the end, there still needs to be a story where the weight of evidence is that a therapy or a biomarker is fit for the purpose with a well-defined purpose. So, you think about the package that needs to go up to make a case when you're up on that stage making that presentation. It's the strength of your evidence. Um, if it's not, if you're using a black box AI approach and you kind of can't explain why you got there, or say, a target identification profile isn't very strong, the safety, are you really going to make safety decisions based on something you can't explain? That's not strong. So we're getting closer. I think AI is part of some of the analytical methodologies. I think that's going to be fine. But again, it's more AI, kind of knowing where to place AI in the entire context of say a package like this is really important. It probably would not be sort of a primary make or break analysis. But then again, you kind of ask what is a primary make or break analysis? You really want to have a clearly defined outcome and what that measure of success would be.
Yeah, well, I mean, I think, going even back to the very beginning of our discussion and the notion of these dynamic biomarkers, because, a topic that Ornit and I have been wrestling with as in this podcast series and in our journal articles, as we move into the next wave of omics integration, particularly with proteomics and transcriptomics, it definitely feels like AI is everywhere. But
Mhm.
all too often the tools being used are they somewhat black boxes. I mean, you and I have seen lots of papers like this.
Mhm.
Inputs go in, predictions come out, and no one really knows why. And yet there is this issue of sort of dynamic data and multimodal data that we have to sort of make sense for. Where does this head?
I mean, I think it's evolving. I think it's evolving to the point where some of the explainable AI methods are probably going to be a bit further along and more accepted. I think some of the pathway analysis that most people crank through will get better, will improve. You just have access to more information. I've seen now some AI methods where there's actual cross-checking of their own results as part of the process. I think that's really pretty key. So it's not, it kind of gets around some of these questions for hallucinations in AI, right? You would not want to make a presentation on a therapy and then say, oh, well, that was an hallucination. But I've seen AI methods that actually are designed to specifically look for hallucinations in other AI methods. So, I mean, I think there's a bit of iteration there that might work.
And Mike, me and you had a lot of discussion about data transparency and rigor in science, and yet your work is consistently emphasized transparency and understanding the full analytical chain, the input, the logic in the middle, and the biological meaning of the output. So the question is, how do we preserve the clarity and accountability as AI becomes more embedded in proteomics and transcriptomic workflows? Is it about model design? Is it about team structure? Or is it about scientific culture? Or any of these combination of these factors?
Right. Well, thanks for the point about transparency. That has indeed been a driving force for me for a very long time. So even as the analysis of omics data gets more complex, there's still an emphasis on transparency expected by sponsors, journals, and collaborators. I think this is an area where technology can help. So things like Jupyter notebooks, for example, easily capture the analysis step. Electronic lab notebooks for analytical and computational studies also really helpful. Explainable AI goes a long way to bring in AI into a more transparent model as well, especially for analysis of high dimensional and multimodal data. Explainable AI has the potential to bring both quantitative and qualitative results together for better interpretability.
Thank you, Mike. And one more thread before we close. As this AI method become more common in the analysis of omics data and multi-omics data set, they are starting to influence how clinician and trial sponsor interpreted finding and make decision and even design endpoints. From your perspective, what will it take to make sure that AI driving insight are not just technically sound, but also clinically actionable?
So to make sure that AI results are clinically actionable, it's best to start with an aim to define what this clinically actionable context is. So is it choosing a dose? Is it repurposing a drug? In the math modeling culture, there was always a feedback loop where experiments provide data for the model suggests new data. New data is used to build a new model. So this type of feedback loop, I think, was very important, and the iteration would continue until you get convergence. I'm thinking that with AI, it may be very similar. So AI models can be continually improved based on new data, almost getting back to this point that I was making a little bit earlier about AI models or some model and eventually humans kind of checking the context of results and saying, well, is this something that's clinically actionable at all? So in a sense, the first set of AI insights may give a very limited set of clinically actionable results. Maybe then refinement of the AI model can happen with more data and more interpretation. So if you just think about the example of the drug repurposing, a first run might provide some insight, but the coverage of the applications is too general to be clinically actionable. It could come down as well. This drug can be repurposed for so many different diseases, it's not even, possible to make something of this. So the insight needs to get refined. You need more data, but again, kind of the same way as that mathematical modeling example, you get more data, you get a new model, you get some new AI results, new experimental results. You may suggest, I think very importantly, suggest new experiments, and then the next iteration narrows down to something that's more clinically actionable, something that's usable with, for the end game, where the end game is really the right drug at the right dose for the right patient.
Thank you.
Mike, this has been a really rich discussion. We've talked about language, data, design, and collaboration, and how science can lose its direction when it treats tools as a purpose.
Yes. And one thing I will take away from this is that the idea that precision is not just about measurement, it's about mindset. That is where people like you, Mike, play such a vital role in Alzheimer's disease research, helping all of us to recenter the science. So thank you for that. And to our listener, stay tuned. In our next episode, we will explore how cognitive and behavioral endpoint, including digital phenotyping, can complement omics-based approaches. Until then, I'm Ornit Chiba-Falek.
And I'm Ara Khachaturian. Thank you for listening.